Hello everyone, today I'm excited to present my research project titled Analyzing the Impact of Vaccine Types on Total Vaccinations in WHO regions. In the midst of the global vaccination effort against COVID-19, we set out to explore a critical question. Does the choice of vaccine types make a significant difference in the total number of vaccinations administered? Throughout this presentation, I'll discuss the problem we aim to address, our data sources and methodology, and the conclusion we've drawn from our analysis. Our project is driven by the following objectives: evaluate the mean total vaccinations administered in regions with varying vaccine types, conduct a two-sample t-test to determine statistical significance, utilize advanced data analysis techniques for precise insights, provide valuable information for optimizing vaccination strategies, enhance our understanding of pandemic response factors. Let's explore the answers together as we navigate our research findings. This is our research question. Is there a significant difference in the mean total vaccinations administered between regions with a high number of vaccine types used and regions with a low number of vaccine types used within the WHO regions? Let's begin by examining our hypothesis, which centers around the impact of vaccine types on total vaccinations within WHO regions. We are investigating whether the number of vaccine types used, categorized as high or low, plays a significant role in total vaccinations. Our null hypothesis states that there is no significant difference in the mean total vaccinations between regions with high and low vaccine type counts within WHO regions. In contrast, the alternative hypothesis suggests the presence of a significant difference. To test this hypothesis, we will employ a two-sample t-test, a statistical method that allows us to determine if the observed difference in mean total vaccinations between the two groups is statistically significant. This analysis will help us discern whether the choice of vaccine types indeed has a meaningful impact on vaccination outcomes. In our analysis, we conducted a Welch two-sample t-test to investigate whether there is a significant difference in the mean total vaccinations administered between two distinct groups of regions within the WHO regions. These groups were categorized based on the number of vaccine types they utilized. The t-value, a key statistic in this analysis, indicated a small difference in means between the two groups. Importantly, a positive t-value in our context suggests that regions with a higher number of vaccine types recorded a notably higher mean total vaccination rate compared to regions with fewer vaccine types. The degrees of freedom for this test were relatively high, a value essential for determining the critical t-value. Our analysis yielded a p-value which, when compared to the commonly used significance level of 0.05, was relatively small. This suggests that we would likely not reject the null hypothesis. In simpler terms, our data and analysis do not provide substantial evidence to support the existence of a significant difference in mean total vaccinations between regions categorized by high and low vaccine types. Essentially, the p-value assesses how likely our observed outcome is if there were no actual difference between these two groups. A smaller p-value, such as the one we obtained, suggests that the observed outcome could reasonably occur if there were no real difference. Additionally, we calculated a 95% confidence interval to better understand the plausible range within which the actual difference in mean total vaccinations between the two groups might fall. The lower limit of this interval was relatively small, while the upper limit was relatively high. With 95% confidence, we can state that the true discrepancy in mean total vaccinations likely exists somewhere within this range. This interval illustrates the degree of uncertainty surrounding the precise difference in means. Based on our comprehensive analysis and a significance level of 0.05, our findings lead to the conclusion that there is no statistically significant difference in the mean total vaccinations between regions with a high number of vaccine types used and regions with a low number of vaccine types used within the WHO regions. This conclusion arises from the p-value being relatively small and insufficient to reject the null hypothesis. The project's findings reveal that the specific types of vaccines used within the WHO regions have a limited impact on the overall vaccination numbers. Although there was a minor variation in vaccination rates between regions employing a diverse range of vaccines and those with a more restricted selection, this variance didn't hold statistical significance. As a result, it suggests that there are likely other significant factors influencing vaccination rates. These results underscore the necessity of adopting a comprehensive approach to address vaccination disparities and enhance the global pandemic response, extending beyond the sole consideration of vaccine types. Thank you for watching my presentation.